Soy el doctor Jaime Ortiz López, profesor de la Escuela Superior de Física y Matemáticas. El doctor Miguel Tufiño Velázquez, director de la Escuela Superior de Física y Matemáticas, se disculpa por no poder estar presente en la apertura de esta segunda y última plática del doctor Gellermann y me dejó encargado que yo abriera la sesión. En el marco de la cátedra Eugenio Méndez de Ocurro 2021, presentamos la segunda y última plática titulada Non-invasive Optical Detection of Carotenoids in Human Living Tissue, presentada por el doctor Werner Gellermann, fundador y gerente general de la compañía Longevity Link Corporation, situada en el Parque Científico Tecnológico de la Universidad de Utah, Estados Unidos. Ayer dimos una versión más o menos amplia del currículum del doctor Gellermann, así que hoy iniciaremos con una versión más breve para aquellas personas que no pudieron estar en la plática de ayer. Me da mucho gusto presentarles al doctor Werner Gellermann, amigo y condiscípulo mío de hace casi 40 años, cuando ambos estudiamos el doctorado en física de estado sólido en la Universidad de Utah, compartiendo el mismo director de tesis. El doctor Gellermann estudió su licenciatura y maestría en el Instituto de Óptica Cuántica de la Universidad de Hannover, en Alemania. Tiene alrededor de 81 artículos científicos publicados, 12 patentes y una marca registrada. Varias de sus patentes involucran el desarrollo de nuevos láseres de estado sólido en el infrarrojo cercano. Además, ha desarrollado metodologías de detección óptica no invasiva para carotenoides y compuestos similares en tejidos humanos vivos y ha desarrollado nuevos instrumentos alrededor de esta línea de trabajo en lo que podríamos llamar óptica biomédica. Las pláticas del doctor Gellermann las está realizando desde Alemania por el momento, donde ahora se encuentra temporalmente atendiendo a su octogenaria madre, por lo cual me ha pedido que el número de preguntas al final se limiten solo a 3. Sin embargo, me ha comentado que pueden interrumpirlo para hacer preguntas en cualquier momento durante su plática. Entonces, damos comienzo. Okay, Werner, please, you can start your second talk, please.
Okay, let me share the screen here. Okay, can you see it?
Okay, very well.
Okay, let me smallerize this here. Okay. Okay, can you hear me?
Yeah.
Very well. Okay, let me start with the second part of my talks here then. And this one is about macular pigment in the human retina. Turns out that the human retina contains carotenoids, the molecules I introduced yesterday. In particular, you have 2 carotenoids sitting in the human retina, and those species are lutein and zeaxanthin. Before I get into that, let me first sketch the anatomy here of the human eye, shown here at the top. Here's the eyeball. From the left side here, you have the light entering through the cornea, and then it travels through some aqueous liquid here towards the natural crystalline lens. And from there, the light falls onto the human retina, which is a hemisphere at the back of the eyeball, okay? And this is a very sensitive optical sensor that detects the light, and it sends the signals from the light detector, from the photoreceptors into the optical nerve, which is here, through the optical nerve into the brain. And then the brain makes sense of all of these light signals received along the retina and generates an image of the outside world, which is upside down. If you look at a little segment here of the retina, shown by this white square here, and enlarge it, you kind of see in schematics, the cell structure. And it turns out that there are some elongated specialized cells which have various functions. Actually, this black one here, which is transparent in reality, is a nerve fiber layer that receives the electrical impulses generated by the photoreceptors in the back here. And then shown here is red, green, and blue arrows. Those are the light-sensitive, color-sensitive photoreceptors that are responsible for the color generation. And then the others are kind of rod sensors that only detect black and white light signals. So the whole arrangement is here all along the hemisphere in the back of the eye, okay? And when you look directly, if you slice through here, along this vertical plane here, then and look directly through the pupil into this eyeball, you see something like this. So the optical nerve that in cross-section is here, appears here. Yeah, it's the optical nerve. And then from there, you have all kinds of blood vessels here going out into the peripheral retina. And then there's a region here, which is relatively free of blood vessels. There's almost no blood flow here, and this is called the macular region, the macula. And then the center of the macula is called the fovea. So the fovea is right here in the center. This is called the foveal pit, it's exaggerated here. And then this whole region is called the macula. And the diameter of this area here is about 2 millimeters only. So it's very small. And this is the area in which all of the color vision of our eye happens. Over here, you have photoreceptors too, but they're only responsible for dark vision. So right here in this 2 millimeter spot, that's where all of the action happens. All of our light vision happens. And it turns out in cross-section here that there's some yellow pigmentation here consisting of lutein and zeaxanthin. And that pigmentation comes from the diet. These carotenoids we eat with fruits and vegetables. And then the body, in some magical way, extracts only lutein and zeaxanthin from this family of carotenoids and then puts it into the retina here, in this area. This is a very specialized process, and it's facilitated by the existence of specialized binding proteins which sit here and catch the lutein and zeaxanthin molecules. How this works in detail, nobody really knows that, but it's still a subject of intense research in the ophthalmology community. In any way, turns out that the zeaxanthin is located centrally, so it sits right here in the foveal pit in the center, and then the lutein is a little bit more peripheral. People know this from experimenting with excised eye cups and extract the carotenoids from there and distinguish between lutein and zeaxanthin. Why that is, nobody knows, but the lutein sits here and the zeaxanthin a little bit more in the periphery. In any case, what's very important is that according to epidemiology inside, there is a strong protective effect of lutein and zeaxanthin. You can think about it maybe as being some internal sunglasses, yeah? So the yellow pigmentation absorbs blue light when it comes through here, and then before it hits the photoreceptors, which are here in this area, the yellow pigmentation filters out the phototoxic blue light components, because they are, as you know, the The blue photons are quite energetic, and they may have enough power, enough energy to photoionize the retina and do some long-term damage. And this is actually the story I heard from my long-term collaborator, Professor Paul Bernstein at the University of Utah Moraine Eye Center, who basically challenged me to come up with an objective optical method to quantify these pigments here, and then maybe develop some instrumentation that can detect them quantitatively. So if you run the absorption spectrum of an excised eye cup, yeah, an excised eyeball, if you dissect it and cut it out, and we stick it in our spectrophotometer, you see an absorption band, which looks exactly like the absorption spectra I showed you yesterday for these carotenoids. You see even here 3 very well resolved vibronic bands, yeah, and then the strength also of this band depends on the concentration. Okay, so in the ophthalmology world, what keeps those doctors very busy is to deal with age-related macular degeneration. So the retina that I just showed you before no longer looks healthy and has some nice orange coloration with these blood vessels coming out, but you see all kinds of growth occurring in the retina. Here's the optical nerve again, and you here see some yellow areas, which are some fatty deposits. And then what happens is that in the extreme case, you have some hemorrhaging, there's some blood occurring in the retina, and then the macular area, which is here, is totally degenerating, and it kind of looks like this, and the people become blind. The reason for this is oxidative stress, very likely, where these photoreceptors here are getting destroyed by some oxidative processes, which I talked about yesterday. So what can we do about that? Before I go into the details, just let me show you the prevalence of the disease. It's the leading cause of irreversible visual loss in developed countries. 200,000 Americans have the advanced form of AMD, and that usually sets in with age of 50 or so. By age of 70, maybe 30% of the people have a mild form or some advanced form, medium form of macular degeneration. So for those people, instead of seeing a clear image here of the world, they see something like this, where in the center everything is deteriorated, the photoreceptors don't exist anymore, and the signals are blocked out. So these people only can see a little bit in the periphery, if they're lucky. So how to measure the macular pigment in the human retina? So quite a few years ago, we set out to do that and try to develop some method which is non-invasive, fast, optics-based, quantitative, and maybe can also catch the spatial distribution of these pigments. Historically, it's quite interesting. In physics, we all heard about James Clerk Maxwell, very famous physicist. He developed the Maxwell equations, the wave nature of light, and actually he saw this already in 1856. So he described a dark spot in the blue region of the prismatic spectrum, as he called it. So the way it works, and if you can repeat this for yourself, is if you stare into some green background light, you probably don't see this here from the slide presentation in PowerPoint, but if you kind of block all of the light in your field of view and stare at a green screen for a minute or so, maybe half a minute, and then quickly switch to blue, you will see that the photoreceptors have been saturated and there will be a dark spot in the middle where your macular pigment is. So you might want to try that after this talk. About 30 years ago or so, 40 years ago, people developed a a psychophysical method to catch the macular pigment, which is described here. In principle, you have a light bulb, and you split the light bulb up into two paths, one green one and one blue one. And then you combine them here with a rotating mirror, and you will look into the light beams sequentially, the green one and the blue one. And then the blue beam is attenuated by the macular pigment, but the green beam is not, because at the green wavelengths, we are outside the optical absorption band. And then what you do is you push in a attenuator, a gray filter into the blue light beam until the intensities appear to you the same from this channel as compared to this channel. And then you know that the macular pigment optical density is exactly the same as the optical density that you push into the blue light beam. Okay, so from that you can determine the attenuation coefficient or the optical density of the macular pigment. So the way it looks in the lab is you have here a box. The box has a light source, two channels, and so forth. And then you look through a hole here at these two light channels, and you dial in the attenuation into the blue light channel here with your hand. And then you determine for yourself when you have a matched light intensity. And then you read off the attenuation here, and then you have the result. This is one of my postdocs. And we did a extensive study, checked out the pros and cons of this technology, and determined that definitely it can be improved. The main reason is that this is a psychophysical method where the result is determined by yourself. You have to judge for yourself when you have achieved attenuation such that the two light channels are balanced. And that's a task which is very difficult to perform for old people. You know, people shake their heads already and cannot see very well and cannot concentrate very well. So basically, it doesn't really work very well for the elderly. So we set out originally to use our Raman spectroscopy first, and then later on we switched to autofluorescence attenuation spectroscopy. And those are the two topics I want to talk about next. So first, objective method is resonant Raman spectroscopy. I discussed it yesterday for the measurement of carotenoids in human skin. We can also use it for the eye, turns out, although people told us we were crazy to shine a laser beam right through your pupil onto your retina, into the macula, which is, of course, the most light-sensitive spot. But we tried anyway and managed to do it in at very low light levels, which are considered safe. And you know, the very strict ANSI standards, safe light exposure standards set up by the government regulatory agencies. And we, of course, went below those intensities and tried, and lo and behold, we could get a signal. So here's the setup. We used an argon laser, attenuated it, of course. Here's a line filter, mirrors, beam splitters, and then shine it through the pupil into the eye. And then the back scattered light from the Raman effect comes back to mirror M2 here, and then is reflected by the beam splitter into a spectrometer. And what you see here is at the bottom is the computer screen. We have here a Raman scattering spectrum, which is imposed spectrum of a strong fluorescence background coming from some material inside the eye. And then if you look carefully on top here, you have these sharp characteristic peaks coming from the carotenoids, the carbon double bond peak, the carbon single bond peak, and even the methyl sideband peak. Then if you subtract the background with a higher order polynomial, do some fitting, then we can extract the Raman spectrum here, and we can quantify the concentration of the macular pigment by the amplitude of this peak here, the carbon-carbon double bond peak. You need to align it correctly, so we provided some target here for the person to adjust the instrument correctly, and once it's aligned correctly, you push a button and you get the result. There's some the method works, but of course it's not too attractive because you still have to use a laser beam for the light excitation. So whenever you use laser beams and shining it into your eye, it's not the most acceptable method. So what we did is we tried some other method, and that's based on fluorescence. So we call this autofluorescence attenuation. And the way it works is, here's again the cross-section through the human retina. So here's again the foveal pit, the macular region. Shown here are the retinal layers, the nerve fiber layer, lower layers. Here's the choroid, which supplies all of the nutrition coming into the eye from the back of the eye here. And then here is again the macular pigment shown as yellow coloration. And it turns out that here in this layer, way back in the eye, you have a pigment which has nothing to do with carotenoids. It's called lipofuscin. Lipofuscin. Lipofuscin is some pigment that's generated by the natural decay of the cells. As you know, the cells grow and die all the time and are replaced. And the lipofuscin is some sort of an H pigment, which is removed by the body as the cells decay. But it cannot be removed as quickly as we age, and it stays behind with increasing age. Babies don't have any lipofuscin in their eye, but the older you get, the more lipofuscin you have. Okay? And what we do here, we use this lipofuscin as an internal light source. It turns out that the lipofuscin has a very strong fluorescence effect. Remember from yesterday, I told you that the carotenoids, when we excite them, they have no fluorescence. Nothing is coming out. So we have to use an indirect way of catching them. So what we do is we shine blue light into the eye, onto the back of the eye where we have the lipofuscin pigments. And then these lipofuscin pigments generate a red fluorescence here, occurring at about 600 nanometers, 620 nanometers in their peak. So that's in the red wavelength region. So red light is coming out of your eye, okay? This is actually the light that you sometimes see coming through your pupil when you have some camera flash. So your eyeball looks red. So we have the lipofuscin light coming out everywhere where you shine blue light into the eye, okay? And then the idea is that we compare the fluorescence here in the peripheral retina with the fluorescence here in the center of the macula. Why? Because here in the center of the macula, where we have the carotenoid pigments, the blue light is absorbed, okay? It's attenuated. That means you have very weak fluorescence coming out here because the excitation light intensity is attenuated. But here, the excitation light is not attenuated, yeah? There is no carotenoid here. So you have very strong fluorescence coming out here. And then the higher the concentration of the macular pigment here, the larger is the contrast between the fluorescence in the periphery and the fluorescence over here. So it's a very simple trick, and the method is easily adapted also for spatial imaging. Here's our first experimental setup for the autofluorescence imaging. This is Mohsen. He received his PhD degree dealing with this long time ago. And this was our first setup. So we look here into the light source, which in the beginning were two laser beams, are now replaced by a blue LED. Blue LEDs didn't exist actually when we started, but now we have a blue LED. So what we do is we shine a blue LED here through this beam splitter into the eye, okay? And then we route the fluorescence coming back out of the eye into a CCD camera. So here we used a camera used normally in astrophysics, very sensitive, cooled CCD array, very high sensitivity that can catch the fluorescence very quickly within a fraction of a second. So that's also a very important requirement. You want to have a method where you take the image, the picture, quicker than you can blink with your eye. So it has to be done in a fraction of a second. So you generate a light flash, and before you can blink, the camera has captured all of the information. Then you display it on the computer screen. So here's some details about the image acquisition and processing. For comparison, here's the white light reflection image for a healthy human retina that I showed you before. So here's the macular region with the lutein and zeaxanthin carotenoids sitting there. Here's the optical nerve. And then what we see in the autofluorescence image through the CCD camera is this square here shown at the bottom, where you can still see here in grayscale the blood vessels here, same as here. And then you see here the macular pigment as an attenuation produced by the carotenoids. Yeah. So here you have a very low fluorescence intensity, and here in the periphery you have a high fluorescence intensity. So that's how it works. And then for the image processing, what we do is we choose little small circles here in a concentric fashion in the periphery, so in this area here, and those are in pixel intensity areas with high intensities, okay? And then we compare the intensities with the pixel intensity that we get right here in the fovea and in other areas of the macula. So here, there, and then we go out radially. And we do this here according to the equation. So the macular pigment optical density is just the density here in the periphery minus the density in the fovea. And we can get this simply from the logarithm of the ratio of the intensities. So it's very simple and can be done very quickly with computer processing. You don't need a fancy computer. You know, there's a lot of horsepower now in laptops and so forth. So you get almost instantaneous results. Okay, now let me show you some results for healthy subjects. Here is a first example where this is the raw image of the autofluorescence instrumentation. And when we convert this grayscale image into a color image, then we get a distribution like this. And then we can color code it, like in exactly the center here. By the red color, we characterize the high intensities, the high absorptions. And then by the green blue colors here, we have the low absorptions. And then you can map out actually the macular pigment in space in real time. Okay, so this person has a macular pigment distribution which looks sort of, we call it like a volcano with a kind of triangular shape here. And this subject here, we say it has a narrow macular pigment distribution and has high central levels. So this is very good example of a person who has a very strong macular pigment concentration. And this person should be very well protected by these pigments in terms of his photoreceptors. Here's another example. In some people, we find that instead of this narrow distribution, they have a very broad distribution, okay, with some sub-peaks. So here it's very broad, some sub-peaks. And overall, the levels are still very high, but there is some missing top here. Okay, and in this person here, very interesting case where you have what we call a ring type macular pigment pattern, where in the foveola, in the center of the macula, you have some relatively high concentration here, but then there is a region here surrounding it where there's almost nothing or very little pigmentation, and then here you have a ring again. So what we see here is that the carotenoid distribution in the human macula in some subjects seems to fragment. So it goes down, and then in some cases we see even the absence of macular pigment, and that's we want to avoid. And then of course the hope is to remedy the situation by having people take up supplements and bring back the pigmentation. As I told you before, people know this from biochemistry. Right in the center of the macula, you have zeaxanthin, so you can do this with the help of zeaxanthin supplementation. And then in the periphery, you can try to bring up the concentration again with the consumption of lutein. Okay, there is a very severe problem with our idea of autofluorescence imaging, and that's the occurrence of cataracts. Okay, as you know, when people get a little bit older, by age 60 or so, what happens is that the lens very often starts to degrade. It becomes a little bit cloudy, the human eye lens, and you actually could become blind from it because it turns brown in some cases, and then you cannot see anymore. So this is a severe problem for any imaging of the retina, because instead of getting a nice clear picture, if you have a medium cataract, it doesn't look so nice and clear anymore, but the whole image gets kind of cloudy. Here's a medium-sized cataract. You can still see the optical nerve and some features here from the blood vessels, even some macular pigment, but overall you don't have very good image contrast. And then it gets worse here in people who have a severe cataract, strong cataract. The whole image is almost washed out. Okay, so here we looked at 74 subject eyes, measured 74 peoples, and measured people before and after cataract surgery. As you know, that's a very common form of surgery where you can go into the eye clinic. They make an incision into your eyeball and take out your natural lens and put in a plastic prosthetic lens within about 10 minutes, and then you have clear vision again. So in principle, that's relatively easy to do. Of course, there are some risks factors associated with it, but it's a very well established surgery, and it's carried out by millions of people. So what we did is we worked with a cataract surgeon at the eye clinic and measured the macular pigment optical densities with our instrument before cataract surgery and after cataract surgery. And as you can see here, each full circle here is the macular pigment optical density for each person. And if you look at the range of macular pigment optical density before cataract surgery, you get an optical density from about 0.45 or so up to about 0.4. Okay, not very large range. But then after cataract surgery, you see the optical densities range from about 0.2 to 0.9, so a much larger range of optical densities, and that's due to this degradation effect of the natural lens. So then how can we get around that? Because we don't want to measure people only after cataract surgery. We would like to develop this technology for everybody at any age who is worried about his eyesight later in life. and want to tell him what his macular pigment optical density is in the presence of cataracts, mild cataracts, because people don't like to get cataract surgery until it's progressed already to some medium stage. But even in mild cataracts, you have when you don't want to get surgery, you have already some image degradation. So what we did is we look at these autofluorescence images quite carefully here, and you see here, if you look at the raw images, here's some kind of nebulous image which is a little bit cloudy. Here's the corresponding image after cataract surgery. And what we discovered is that if we look at the pixel intensity histograms, which is basically counting all pixels of the camera chip that have the same intensity, then we see a very strong difference in the pixel intensity histograms. So that means that if all pixels have the same intensity, you get a very sharp distribution here for the histogram. Yeah, imagine that in the extreme case, you have a totally washed out image, then all the pixels of the camera would see the same intensity. Let's say you have 2 megapixels, they all would have the same pixel intensity of 100 here. If you have clearer images, then some pixels see a low intensity, some people see a medium intensity, some pixels see a high intensity, and then the histogram becomes much broader. So in other words, there's much more information in an image like this. Yeah, you have some darkness here, but you also have some very strong light intensities here, and correspondingly, the pixel intensity histogram has a much larger width. So then we had the idea that we could use the feature of these pixel intensity diagrams as a measure for the grade of the degradation. So here, this has a very low, a relatively small base width of the pixel histogram, and this one here is much larger. So maybe we can use the base width of the histograms as a measure, as a quantitative measure. Okay, so then if we plot for all of these subjects that we measured in the eye clinics, the ratio of the macular pigments after surgery and before surgery, which is shown here, yeah, it can go up to factor of 9. So in subjects with very bad cataracts here, the macular pigment optical density is attenuated by a factor of 9. If you don't have any cataracts, then it's blocked by a factor of 1. That means it's not blocked at all. Okay. And what we saw here is it nicely follows on average a parabola here. Okay, with a high correlation coefficient of 0.89. That means we have a pretty good handle on the image quality of the images that we capture based on the base width of the histogram. Yeah, so there's a parabolic fit, and then now we can use this fit as a correction lookup table in our software and multiply the macular pigment optical density that we determine for each subject. So if the subject has a very strong histogram base width, then we don't need to do much correction. We don't need to multiply it much, but like here for mild cataracts already, we need to multiply it maybe with a factor of 3 on average and then go up. Also, we could use this technology as a byproduct. We could use the image clarity or the pixel base width, the pixel histogram base width. We could use it already as a indication for the existence of cataracts. Yeah. So it's very easy. We just take a picture. We look at the quality and tell the person whether he has a clear lens still or not. All right, then we set out to develop a commercial prototype based on a fundus camera. The fundus camera is an instrument which is used in ophthalmology for 100 years already or longer, and is used is kind of an all-round instrument to look at the general properties of the retina, to check whether there's some holes in it, whether there's some bleeding, or whether people have some abnormalities, or whether everything looks okay. So these cameras have emerged into some high-tech, sophisticated cameras over the last 100 years. And every so often, there's some improvement in light sources, in detectors, in aiming beams, in computer processing, and so forth. So these are very modern, high-tech instruments supplied by various companies already. And what we did is we got hold of a camera, a fundus camera, actually collaborated with one of the commercial suppliers for cameras, a company from Japan called Nidec. This is the optical layout of the camera. Here's your head, here's your eyeball. And then what you do is, in most cameras, you have a halogen lamp. The light is filtered here, or you have an LED. You can switch between the two. You can bring in excitation light filters, whatever you want. You have some slit stops here, and then the light is routed through magnifying optics and so forth to some mirror, which has a hole in the center, and is routed here to a very large objective lens into the eye. So it's focused onto the cornea here, goes through the pupil and spreads out here. And the light that comes back, the reflected light, goes through the whole mirror here, is magnified and imaged, corrected for astigmatism and whatnot, and then is caught by a camera. And what we did here in our system, we replaced the normal camera here with a autofluorescence imaging camera, the camera I had introduced earlier. And also what you can do in these cameras, you can provide some aiming beams like a red LED. You send this here through a focusing charge, and you have your subject look at this beforehand. And also the operator can look at it through an additional optical channel here and make sure that the eyeball is well aligned before you push the button and generate the light flash. The image processing, again, is done with our software in this way here. So the operator chooses a pixel locations here, depending on the image. Make sure that there are no obstructions here, no bleeding or no abnormalities in the retina. Yeah, picks a couple of reference circle here in the peripheral retina. And then the computer automatically calculates the what we call a line plot here, which is the macular pigment optical densities derived from these contrasts, running through a nasal temporal line through here, right through the middle of the distribution, and then plots out something like this. So this is a line plot of the macular pigment going right through the center here, and then you can read off the optical attenuation, and from that the concentration right in the middle, which in this case is about 0.8, very high, and then it drops off here to the side. Also, we can display the spatial distribution, and we can portray this in colors, as I showed you before, and so on. Here's the experimental setup. These are my collaborators here, Maya and Igor. They've been working on this topic for a long time. And so what you do with this camera is you, once you have alignment, you push a button, and then you have a what we call a soft blue LED flash generated by a blue LED. And it's we managed to cut the flash time down to about 100 milliseconds, which is very short, much faster than you can blink. So then the eye, the retina here, is exposed with this blue light flash, and then almost in real time you can look at all the line plots, the MPD levels from the line plots, the 3D distribution. And you can also integrate over the whole area and determine the macular pigment volume. And what we hope to do now is that we can use it for rapid eye health screening of large populations. And what we try to do is now get the interest of companies that provide dietary supplementation. There are several companies out there already and selling macular carotenoids, lutein, and zeaxanthin. And what they could do with technology, with this technology, is to prove that their formulas work, and then they can use this as a differentiator in the industry. Okay, how am I doing on timing here?
It's running 54 minutes.
Okay. If you want, I can still talk a few minutes about our latest technology in the skin world.
Oh, okay. Yeah, maybe.
Or I can take questions at this point.
Oh, okay. Let me ask
As you like it.
the audience just a minute. ¿Hay alguna pregunta que puedan hacer en este momento de todo lo que han presentado respecto al método, a la tecnología para el análisis de la mácula y de lo que ha presentado hasta ahora el doctor Gellermann? I have my own question.
Okay.
Okay. Is it, how come only 2 of the carotenoids are the ones that are sitting in the macular fovea? Because
yeah, that's a very good question. Whenever you have a selective uptake of something like this, then people know that there must be some binding proteins involved. So the way it works is that these huge proteins, they have some pockets, and these lutein and zeaxanthin, they have some OH groups sitting in their end rings of the carbon chain, and they fit into this pocket.
Oh.
It's kind of like a key and a lock situation. So these 2 fit in there, and all of these other molecules that are coming by, they're not bound by the protein.
Okay, I hear there's another question in the chat. Jesus Adrianes Corsa, how long time have you been working in this, and what has been your inspiration in this work, in this line of work?
We've been working on this for probably 20 years, starting with exercise eyes, and did a lot of other things which I didn't talk about, and worked with new technologies, new computers, new light sources, and ultimately try to commercialize these technologies, both in the eye world and the skin world. It's a very long process. First, you need to develop the optical concepts. You need to generate patents to protect your ideas. Then you need to run clinical trials. You need to do these clinical trials at our local eye clinic, and other people want to also be convinced that it works. And then in the end, you need to have a commercial instrument, and then you need to also obtain government approval, FDA approval for that. So this whole time span before you can come to market with something like this easily takes about 10-15 years if everything goes well. In the beginning, we used Raman spectroscopy. Today, I talked mostly about autofluorescence imaging. That turns out to be the best way of doing it. It took us several years to figure out what works best, what does not work best, to also compete with other groups who push the flicker technology. So it's a very complex environment.
Another final question before you continue. Would it be more convenient to use a continuous light source like a xenon lamp when you are exciting the eye, so that you can maybe even have a continuously let's say excite, and then you maybe find the optimal resonance response instead of using a laser?
Well, in this technology, we don't use a laser. We use a blue LED light source. Yeah, we call it a soft blue LED flash. So it's basically a blue LED that you see all of the time now in lighting.
Yeah.
Applications. Yeah, you probably have one in your living room. A blue LED, shining from your computer indicator light or whatever. Something like this, and we just flash it for 100 milliseconds. So it's a very benign light source. It's not very strong. We have a very sensitive detector, so we can manage to get this with a very weak light source. And what is very important, we need to do this for a very short time, 100 milliseconds, so the eye doesn't blink. Also, what is very important here in our application is the concept of non-Madriatic measurements. What that means is normally when you shine light onto the eyeball, the pupil will constrict. Yeah, and you avoid that with a flash. So before the pupil can constrict, you have already taken the picture. So if you look into a continuous light source, then the pupil will narrow and a good imaging will become more and more difficult. That's why you go into the eye clinic for some regular measurements. What people do there is they will dilate your eye. So the pupil will lose its ability to constrict and become large. That's called mydriasis.
Okay.
And what we want to do here is we want to measure people in its normal eye state, avoiding pupil dilation. Yeah, we want to develop this as a screening technology for large populations where you go into some regular optometrist shop where you buy your eyeglasses and set up this instrument there and have people screened without getting dilated in their eyes. So there are many reasons for going with a very fast light flash.
Oh, okay. Gerardo Contreras, ¿quieres hacer pregunta?
Yes. Good night, Werner. Question. Although the photo-optic sensibility of the eye is okay, by the handbooks, they gave you these photo-optic effects. This is a technical question. How did you determine when laser, I suppose years ago, the low light intensity to put on the macula, or to track the macula? How do you determine it? Because the sensibility of the eye for each person is quite different, a little bit different, not so much. The photo-optic effect gives you the maxima around, if I'm not wrong, around the 500 nanometers. What was the determination? Although you have already changed with the LEDs, you are not using now laser.
Right, yeah, it's a good question. With the laser technology that we used in the beginning, we could only also measure a spot. Yeah, we could not measure an image. We could not get the spatial distribution. In order to do that, we would have to increase the laser power a lot. So there are a lot of disadvantages with this concept. In terms of the light levels at the time, there are some light levels prescribed by the regulatory agency called ANSI. That's the agency that determines at which wavelength you can have how much intensity. So we simply measured the power of the laser beam and then calculated the intensity that we would have at the back of the eyeball after expanding it to about 2 millimeters. And then we made sure that we were under the safety limits, which are established in the in some tables supplied by the regulatory agencies.
Okay, okay. Thanks a lot. Thanks a lot, Werner. Yes.
Sure.
Okay, but then do maybe continue maybe 15 minutes more.
Okay.
Okay.
Very well. Recently, so this is going back. So this technology is basically waiting for some large company that comes to us and basically purchases the technology, purchases our patents. We have good patent protection on this, and it's a company that's willing to go out and supply an instrument like this to the optometry community. In the meantime, we developed further our skin tissue measurements based on a new spectroscopy. That's reflection spectroscopy. Yesterday, I talked to you about Raman spectroscopy, for which we did most of the applications in the medical field. But recently, we further miniaturized the technology. We're always trying to do that as new componentry comes onto the market, and try to make it smaller and smaller and faster and so forth. So this one here is based on simple reflection spectroscopy. The way it works is you shine a light beam, I0 here, onto the tissue, and then you look at the light coming back. And very simply, you look at the reflection coefficient, R equals I divided by I0. And from that, with a modified Beer-Lambert law here, you can get the concentration of the absorbers inside the skin if you do it right. So this would be less expensive, it would be faster, and actually it would be self-calibrating as well. So let me show you how that works. We call this pressure-mediated spectroscopy, reflection spectroscopy of human skin. And the idea here is that, as you know, we have in the visible wavelength range, when you look at your, let's see, maybe I even have a slide here. No. If you look at your skin, a very strong color contribution to your skin is provided by the blood. And the blood is so strong that it kind of overlaps the carotenoid absorption, which is weaker, is yellow. So what we do is we push the blood away a little bit by squeezing the skin a little bit with topical pressure. We have a spring-loaded clip, and then we squeeze out the blood here, that we bring under the contact lens here. Then what we do is here from the light source, we catch the reflection of the light and route it into a spectrograph and quantify the signals again with a fast CCD camera. So push the blood away, use white light LED instead of a laser. That's very convenient now because we have very strong white light LED sources. They're all over the place right now. They only cost a few dollars. You can buy them as headlamps. You can mount them into flash lamps now, and they can be very strong if you want. And it turns out we don't actually need very strong light sources, and we can very conveniently use a white LED here to get what we want. And the spectral range is also quite large. It goes from 350 nanometers to 850 nanometers. And what we do is we measure the palm of the hand or the finger, just like earlier with Raman spectroscopy. What you see here is in reflection is the red curve here is the curve that's the reflection of the light, white light coming back from the skin. It looks like this over the whole wavelength range from the blue to the near infrared. And you see here some dips in the reflection here where you have a dip here in this wavelength region and here in this wavelength region. And that comes from the absorption of blood here, okay? It's a very characteristic optical signature of oxyhemoglobin sitting in your epidermis. So it's here. These are the characteristic oxyhemoglobin peaks. And then here is the absorption coming from the carotenoids. And then what we do is we push this away. And then afterwards, at some very modest pressure, provided by some spring-loaded lid of the instrument, we have the blue curve here, where this now is the absorption here of the carotenoids. And that's now done without any interference of the blood, which starts to occur already in this wavelength region. The blood does not only absorb here, but actually also very strongly over here in this range. So it's very, very, very important to push it out. Here's a commercial instrument then, what we call the Vegemeter, since all of these absorptions come from the fruits and vegetables. It turns out we only need about 10 seconds of a white LED exposure. We detect the diffusely reflected light with a spectrograph, and then we spectrally deconvolute the spectrum, and then we can calculate the carotenoid levels. Also, if we want, we can calculate the residual hemoglobin and also the melanin levels, which pop up in the spectra too. So this is how on our computer screen the spectrum looks like in the beginning. The carotenoid absorption is here, not much to be seen because it's totally overwhelmed by the blood, okay? And then we push it away in real time. And as you can see here, we get this condition here. So the blood is very low now. And here we have almost the undisturbed absorption of the carotenoids. Here's some rapid screening. We usually go to trade shows, show this around here. This was in Chicago a few years ago. We measure a couple of hundred people in one day, no problem. And, you know, you can get a very nice histogram for these large numbers of subjects. This is done here by our collaborator in Japan. He measured 570 people here. So again, you have a very nice distribution here. And then what you can do is you can kind of compare the histograms, the average scores of people in various ethnicities and so on, and get some conclusions from that. One interesting fact, for example, is in Japan, where my collaborator there, who is also a retinal surgeon, he saw that when he measures about 1,000 people, the average score is around 350 for their carotenoids. When we compare this with people here in our eye clinic, we get 300, a little bit less. So Japanese are maybe 10% better in the carotenoid world. And then here, there's some other collaborators in East Carolina. They had a Vegemeter in a convenience store, a corner store, where a lot of people go shopping for some processed food, and there the average score was 225, so quite a bit lower. And of course, these people, you know, have a very high risk for developing diabetes and other problems. So quite a significant difference between ethnicities and different people. Here's an application in a Japanese grocery store. So the Japanese are very interested in this. And you see here, they put up one of the Vegemeters right next to the vegetable selection in this grocery store. They put it here in a little pedestal, have some promotional poster around it, and educate people in the importance of fruits and vegetables this way. And here they generated some first data in that grocery store showing how their levels go up on average if they buy and consume more fruits and vegetables. Future developments, we would like to further miniaturize this. We can interface it easily to the cell phone. We have done this already. Also, the footprint itself of the instrumentation can be made smaller. Here's a finger clip that looks similar to the pulse oximeter. You push your finger in here and we can interface it with a PDA device, which is a photodiode array. Yeah, we don't need a fancy spectrometer. We can also try to do it with photodiode arrays. So then the whole system would be about this big. And then ultimately also question is, we had some contacts from some smartwatch companies that approached us and asked whether it's possible to integrate the technology into something like the Apple Watch or Samsung smartwatch. As you know, there's a lot of interest in personal health and wellness and so forth, and people would like to get as much information as possible out of these smartwatches. You can measure already with a modern smartwatch, for example, the heartbeat. They have some green LEDs in here that measure the reflection signal from the skin. And some very modern smartwatches also, they have some electrodes built in already where you can measure the pulse rate, the heart rhythm, and so forth, and even display the envelope of the heartbeat on the screen. So maybe in the future might be possible to integrate this thing in a clever way also into some smartwatch. Okay, so in conclusion, I would say I hope I could convince you that the optical spectroscopy can be suitable for antioxidant research in medicine and may also have some commercial applications in personal health space. And maybe you should eat your vegetables. Okay, here's a list of various groups that collaborated with us on all of these clinical trials. A lot of medical folks here involved. I don't want to go through all of them, but of course, all of this work was not done just by ourselves, but by various collaborating groups. If you have any questions, feel free to send me an email. Here's my email address. And if you want to have PDF files of our publications, then let me know as well. So thank you very much. And if you have any more questions, I'll be happy to answer them.
Okay, thank you very much, Werner. Si tienen más preguntas, ya estamos terminando, ya se termina la presentación. Si hay más preguntas, a lo más unas 3 preguntas. I guess nobody.
Yo puse en el chat.
¿En el chat?
Sí,
No veo.
sí. Antes del profesor Contreras.
Yeah, Gabriela Rueda asked, translating from Spanish, what was how was the process of acceptance of your equipment, your instrumentation from by the medical people, if they accepted very easily, or they were some sort of difficulties?
Yeah, very important question. Of course, in the beginning, they were quite skeptical. So that's why we had to generate all of these publications and work with established epidemiologists and biochemists and medical folks, medical researchers in the field. They would never believe me as a physicist because I don't know anything about their field. So strategically, we collaborated with these folks. In particular, Susan Main from Yale University, she was very instrumental in convincing the nutrition researchers that this would be a good tool to have for them. And it turns out that it's certainly better than the surveys that they had before, where they analyzed the dietary habits of the subjects with food frequency questionnaires. And it works very well now. We have already sold our Vegemeters to more than, I would say, about 130 universities throughout the world.
Oh,
And since about maybe 2 years ago, these people also start generating publications in the scientific literature. So by now it's very well established and it's still growing.
yeah.
As you can imagine, all of this takes a long time. People need to be convinced. They need to apply for funding. They need to buy the instrumentation. They need to get institutional review board approval to measure subjects in their communities or at their clinics, and then they need to publish. So the whole process takes years.
Oh,
But we are past this point right now, and it's very well established, and we get quite a few orders.
good, it's impressive. Gerardo, ¿tenías otra pregunta? Contreras. No, I guess not. Okay. Well, we're very thankful for your 2 talks. It's very interesting topics. I think this would maybe inspire some people in our institute to try to maybe go into similar topics, maybe, and even interested people in commercially acquire your technology. If you, I guess in Mexico there's no distributor of of your technology, I think.
No, we ship those devices ourselves all over the world.
Okay. Okay.
If you like, since we have a good history, we can
So
also
to make
loan you an
contact-
instrument.
Okay. So to make contact or make maybe to buy your instrumentation, you have a link or a sales.
Yeah, you can send me an email to my Gmail address or also Longevity Link Corporation address. No problem.
Okay.
Just send me an email to my Gmail address. And
Okay.
if you have any medical collaborators, they might get some ideas. We see this a lot. In the beginning, very often, we loan them an instrument and then they generate some pilot data. And then they go out and use those pilot data to apply for some funding from some government agencies.
Oh, good information. Yeah, just to finalize, our institute would like to acknowledge your presentations and maybe send you some kind of how you say, well, some presents, small presents for you. I will contact you by Gmail, by the email, so that you can tell me if where can we send you a small package. And when you go back to Utah or while you are in Germany. So
well, thank you very much, but that's not really necessary. They're happy to do this, and hopefully we can one of these days meet in Mexico again after this Corona episode has faded away.
Oh yeah, sure. Yeah, have you been in Mexico in this all 40 years that we?
Oh yeah, I've been there maybe twice, but once in the Yucatan and Let's see, where else? Yeah, I think I've been twice in the Yucatan,
Oh, good.
but not much actually
Yeah.
in 40 years.
Okay. Okay. So, well, I have to close that now. The connection. We are very thankful, but now we are still in contact.
Yeah.
And then we will look forward to meet you again sometime when this problem of the epidemic is gone. Or
Okay, Jaime, it was fun to see you. Okay,
okay,
thank you.
thank you very much. Bye-bye.
Bye, Jaime.